Good day, everyone. My name is Willan S. Duran, and I will be the presenter for the ion exchange chromatography and the high performance liquid chromatography. Before anything else, let me present to you the content of this presentation. First, we will review and recall the previous types of chromatography, which are the size exclusion chromatography and affinity chromatography. After that, we will then discuss one of the main topics of this presentation, the ion exchange chromatography, and we will go through its definition, common properties of all ion exchanges, its principles, and applications. Lastly, we will also discuss one of the major topics of this presentation, the high performance liquid chromatography, and go through its definitions and one of its principles, the reverse phase HPLC. Before proceeding to another chromatography, as what I've said earlier, let's review first what we have learned from the previous types of chromatography. First up is the size exclusion chromatography. We have fully understand and know that the first type of chromatography is a technique that is being used to separate the dissolved macromolecules by size based on their elution from columns filled with a porous gel. As you can see in your screens, the beaker here contains a mixture of molecules that is being poured in the column. And on your left side, there is a representation of how it looks or how the porous gel looks. These mixture of molecules will then be separated because there will be pores that will be opened to trap the smaller molecules inside so that the larger molecules will be the first one to migrate faster and be eluted. However, those smaller molecules will be trapped and will be slowly eluted. The only problem here is that there will be long process for the smaller molecules to be eluted. So a very careful observation must be done in order to determine how these smaller molecules were able to elute. Next, we also know that people who use size exclusion chromatography will be able to understand protein oligomerization, aggregation, conjugation, and conformation. One of the reasons also that analysts use size exclusion chromatography is because they can control polymer strength, toughness, and performance. And lastly, persons who use this type of chromatography can characterize molecules such as proteins, synthetic polymers, and natural polymers from each other, so they can differentiate also. Next, our knowledge doesn't limit only in what we have learned in the first type of chromatography. We also learned that affinity chromatography is a type of liquid chromatography for the separation, purification, or specific analysis of sample components. It also utilizes the reversible biological interaction or molecular recognition called affinity, which refers to the attracting force exerted in different degrees between atoms which cause them to remain in combination. For example, as you can see on your screen, there are three columns present. In the first column, a substance will be poured inside the column, and the sample mixture of proteins will then bind to the substance called the substrate. This substance is attached to the column matrix. So, they will be attached in the column matrix. And the the mixture of proteins will be separated into two. First is the P1, which is the protein that is bind to the substrate, will stay, and the other proteins that were not able to bind to the substrate or the substance will be eluted or removed from the or washed out from the solution. And the protein that is being bind to the substrate will need a high concentration of substance for it to be eluted. There are also major applications that includes or being observed in the affinity chromatography. One of it that includes the separation of mixture of compounds, removal of impurities or impurification process, detection of substrates, investigation of binding sites of enzymes, detection of single nucleotide polymorphisms and mutations in nucleic acids, and many other major applications that fits the very definition of affinity chromatography. Now that you have learned, understand, and recall everything from the previous types of chromatography, I assume that you inculcated in your minds the importance and applications of the first two types. So now, let's proceed to another type of chromatography, which is the ion exchange chromatography. If you have read your book, the definition tells you that ion exchange chromatography is somewhat similar to affinity chromatography. The reason is because it also uses columns that binds the protein to separate it to other molecules present in the column. After the bind protein is eluted, this type of chromatography exploits the interaction between charged molecules in a sample and oppositely charged moieties or part of the molecules in the stationary phase of the chromatography matrix. Here is a sample of the ion exchange chromatography. Ion exchange chromatography is also used in the separation of charged biomolecules. The crude sample, which is the mixture of amino acids, is used as the liquid phase. When it passes through the chromatographic column, molecules bind to oppositely charged sites in the stationary phase. So the negatively charged particles or molecules are being attracted to the cation surfaces. Cation exchange chromatography retain positively charged cations or the remaining positively charged amino acid mixture of an amino acids because the stationary phase displays a negatively charged functional group. Basically, ion exchange chromatography is based on the attraction between opposite charges. The stationary phase is selected such that it has a particular charge. Here, as you can see, this is the stationary phase. There is a particular charge here and it is positive. That attracts the sample molecules. So the sample molecules are the mixture of amino acids. And the negatively charged amino acids or mixture of amino acids are being attracted to the stationary phase. The stationary phase is selected such that it has a particular charge and attracts the sample molecules with the opposite charge. The stationary phase is also known as the ion exchanger. So this part here that process or that attracts the negatively charged sample is also known as the ion exchanger. The process of this chromatography can be represented into four steps. These are the four steps. The first step is the equilibration of the stationary phase to the desired start conditions. The pH and the ionic strength of the sample buffer are selected to ensure that when the sample protein is loaded of interest, binding to the medium as many impurities are possible, do not bind. Therefore, the same buffer is used for the equilibration of the stationary phase. When equilibrium is reached all stationary phase, charge groups are bound with exchangeable counter ions such as chloride or sodium. So for example, if this is the column, the positively charged stationary phase here will attract all of the negative charge samples. If we have a mixture of positive, positive, negative, negative, positive, negative samples, all of the negative samples will be attached to the stationary phase. So, no negatively charged will be able to pass through the chromatographic column. The only molecules that were able or are able to pass through the column are the positive samples or positively charged samples, and they are being uh, eluted, or they will be undergoing the process of washing or elution. Next, the ion exchanger can be of two types: the cation exchanger and the anion exchanger. As you can see here, there is a good representation of that. Cation exchanger has a negative charge, so its structure, the cation exchanger, has a negative charge in it. Its structure contains a negative charge and attracts positively charged cation. So, if this cation exchange were being used in a column, all of the positively charged samples will be attracted to them. And the negatively charged samples were will be able to go through the chromatographic column, while an anion exchanger is positively charged and attracts negatively charged anion. So for this, the previous sample explains it for the anion exchange media. The If this is the column, these are the positively charged anion exchange, and it attracts the negative charge samples. That's why the positively charged samples or molecules were able to pass through the chromatographic column. We also know that there are some properties happening in the ion exchange chromatography, and one of the most common properties of all ion exchanges are: it is almost insoluble in water, and organic solvents such as benzene, carbon tetrachloride, ether, and etc. They are also very complex in nature. So meaning they are often difficult to understand. They are hard to understand. You need to have a better understanding for you to get it, for you to fully grasp the idea of the ion exchangers. In fact, they are polymeric. An ion exchange resin has a ligand with a positive charge or a negative charge. A negatively charged resin is a cation exchanger, and the positively charged one is an anion exchanger, just like what I showed you previously. Ion exchange chromatography also consists of principles. It relies on the attraction between opposite charge stationary phase or the ion exchanger and analyte. It depends on the cation and the anion exchanger. And it also consists of an inert support medium coupled covalently to positive or negative functional groups. Next is the applications. For the applications of the ion exchange chromatography, the main purpose is the separation and purification of charged biomolecules such as polypeptides, proteins, polynucleotides, and nucleic acids. Ion exchange is most widely used chromatographic method because it has a widespread applicability, high capacity, and simplicity, and its high resolution are the key reasons for its success as a separation method. And ion exchange chromatography is widely used by analysts. So, we are already done with the ion exchange chromatography. So let's proceed now to the last type of chromatography, the high performance liquid chromatography. HPLC is a form of column chromatography that pumps a sample mixture or analyte in a solvent, known as the mobile phase, at high pressure through a column with chromatographic packing material. The sample is carried by a moving carrier gas, steam of helium or nitrogen. HPLC has the ability to separate and identify compounds that are present in any sample. So, for example, if we have a sample here, I wait. If we have go if we have a sample here, for example, we have a sample here. This sample will be able to separate and identified using the HPLC. But how? That's the question. How could HPLC be able to identify and separate any sample that can be dissolved in a liquid in trace concentrations? HPLC is used in a variety of industrial and scientific applications such as pharmaceutical, environmental, forensics, and chemicals. So most of the time, this is being used by most professionals. So the principle of this uh, HPLC is that a reservoir holds the solvent. So this is the mobile phase, the reservoir. A high pressure pump will be the one to use to generate a meter or a specified flow rate of mobile phase, typically milliliters per minute. And when the sample goes through here from the mobile phase reservoir to the pump, it will then reach the injector. This injector will able to introduce the sample into the continuously flowing mobile phase. So from here to there, it will continuously go up here up until it reach to the HPLC column. The HPLC column contains the chromatographic packing material needed to effect the separation. So here, this here will be the place where the separation and the where will be the separation is happening. When the separation is done, a detector is needed to see the separated compound bonds as they elute from the HPLC column. Most compounds have no color, so we cannot see them with our eyes. So when the sample reaches the detector, a computer or a chromatogram will determine its uh will determine its uh what it, will determine the sample from the detector. And when the mobile phase exits the detector and can be sent to waste or collected or desired, the the mobile phase that contains a separated compound, which HPLC provides the ability to collect this fraction of the eluate containing that purified compound for further study. So there will be some samples remaining here that will be used for further studies. And these sample here are the ones that were not removed from the HPLC column. And lastly is the reverse phase HPLC. This affirms that the separations and reverse phase chromatography rely upon the reversible adsorption-desorption of solute molecules with changing levels of hydrophobicity to a hydrophobic stationary phase. In reverse phase HPLC, a solution of nonpolar compounds is put through a column that has a nonpolar liquid to immobilize on an inert matrix. A mere polar liquid serves as the mobile phase and is passed over the matrix. The solute molecules are eluted in proportion to their solubility in the more polar liquid. Overall, HPLC is a sophisticated chromatography technique that gives fast and clean purifications than other types of chromatography. But the problem is that it cannot provide you with the credibility that your results are really credible. That's all, and thank you for listening. I hope you understand and learned something about the remaining two types of chromatography.